Great. Well, it's really an honor to be able to be here and give this talk and to be nominated for this award. I really appreciate the recognition. Yeah, my name's Bridget, and I'm currently at the University of California, San Francisco. This work, though, was done during my postdoc in Paula Arlada's lab. So before I talk about the science, I just want to thank the incredible team of scientists that made this work possible. All right, more to come later on that. So here's the objectives of the talk today, to understand the neurodevelopmental consequences of maternal inflammation and to identify cells and gene transcriptional changes mediating the brain's response to maternal inflammation. So I'm a neuroscientist, but also a pediatric neurologist, and I really got interested in this topic through patients I had seen and studies I had read, looking at the relationship between maternal inflammation and infections and adverse neurological outcomes in the fetus and the child. And this relationship is been demonstrated in many epidemiologic studies, but is very poorly understood as far as the mechanism and what we can do about it. So here on the screen, I've listed just a few examples. Elevated cytokine levels, maternal fever, for example, all associated with adverse neurological outcomes in the offspring, including, for example, schizophrenia and autism. And while in certain cases we can treat specific pathogens, we can't do much about the sustained inflammatory response and the effects on the fetal brain. So that's what ultimately I hope we can change. So what we do know is that in the setting of maternal infection or inflammatory processes, that there is a cytokine cascade that occurs, pro-inflammatory cytokines that are upregulated in a pregnant person, and that this results in alterations to fetal brain development. So the question that I sought to answer, and that we hope we got a little closer to with this paper, are what are the cell types and molecular pathways that mediate the response of the fetal brain to inflammation? So we, of course, hypothesized that microglia must be important in this response. Microglia, as you know, are the resident immune cells of the developing brain and the adult brain and have many, many functions that have been demonstrated or are currently being explored in the developing brain. And we took a first look at this question to try to gain some evidence that supports our hypothesis. And this was by examining an existing single cell expression atlas of the developing embryonic neocortex that was generated by Danny DeBella and others in the Arlada lab. So to generate this expression atlas, what was done is that the developing somatosensory cortex was dissected out on a daily basis during development, and then cells were dissociated and single cell sequencing performed in order to look at changes in gene expression over time in a variety of different cell types in the cortex. And what we did is we computed a gene module score looking specifically at cumulative cytokine and pathogen receptor expression during brain development. And of course, not unexpectedly, saw that microglia, shown in green, here had the highest module score. So again, supporting this hypothesis that we wanted to test. So what is the role of microglia in the response of the developing brain to inflammation? To ask this question, we next turn to the maternal immune activation, or MIA model, as a model for in utero inflammation. And this model has been very well described and characterized by many groups over the years. So I'll tell you what we did to induce maternal immune activation, which was injecting poly-IC, which is a double-stranded RNA viral mimetic, basically mimics a viral infection, but doesn't cross the placenta. We injected this at embryonic day 12.5 into a pregnant mouse. And what other groups have shown is that this results in offspring that have alterations in brain structure, in synapse formation, in neuronal gene expression, and importantly, that the offspring that have resulted from this pregnancy have abnormal social behavior that's thought to be autism-like. So I think this is the best model we have to ask this question. So we did, I think, the most intuitive experiment that we could, which is we induced inflammation with microglia present and with microglia absent, and then we looked at the response of the rest of the cortical cells. So to do this, we used a knockout mouse, the CSF1R knockout mouse. CSF1R is a receptor required for generation of microglia. And so as you can see here on the right in knockout mice, there's no microglia populating the embryonic brain. There is a little bit of a caveat, which is that ultimately this is lethal in young pups. And so the way that we performed this experiment was by crossing two heterozygous mice together, injecting poly-IC into the female mouse, and then ultimately selecting out the null pups and the wild type pups for further characterization. We dissected the somatosensory cortex at E15.5, so that was 3 days after the immune stimulus, and performed single cell sequencing, or in fact, single nuke sequencing, as we had to freeze the tissue in order to perform this genotyping. So what I'm showing here is a set of differentially expressed genes after MIA and single cell sequencing. These are log fold change plots, and I'm showing first just the results in APs, or apical progenitors, then I'll show the rest of the cells. So what we noted, looking first on the column on the left, is that there were many genes in the wild type condition in apical progenitors that were either significantly upregulated due to MIA, these are shown in red, or significantly downregulated, and these are shown in blue. And quite remarkably, this is the just coolest finding to me of this whole paper, actually, is that in the microglia knockout condition, these changes are nearly eliminated. And this ended up being the case across a wide variety of cell types in the developing brain, progenitor cell types and immature and more mature neurons, that microglia seem to be required for this normal response of the rest of the embryonic cells to immune stimulus. So of course, we went on to do gene ontology analysis of the genes that were significantly up and down regulated. I won't go through the details, as there's many lines here, but to summarize our findings broadly, and I'm happy to answer questions later on this, we saw that MIA led to microglia-dependent upregulation of genes involved in neuronal fate commitment and related processes, and to microglia-dependent downregulation of genes involved in a variety of cell energetic processes, proteasome synthesis, and also regulation of the cell's response to viruses, which was really interesting. And I'll note that I was excited to see that our findings recapitulated some of the findings in a different paper that was published over the course of our work, looking just at how the embryonic brain responds to immune stimulus outside of the context of a role at microglia. So how does this happen? How can microglia do this? We realized we next needed to turn to looking at microglia specifically, which was very hard to do in the single nuke sequencing experiment, as microglia, you end up getting very few of the microglia actually available for sequencing because they're kind of outcompeted by all the rest of the cells. So we had to look specifically at microglia, and to do this, we performed similar dissection of the somatosensory cortex at a variety of embryonic ages. We just first wanted to kind of understand a baseline before perturbing the system. And then we performed a cold non-enzymatic dissociation of the tissue and an antibody-based FACS purification of microglia in order to generate a normal atlas of embryonic microglial development. And we had a lot of really fascinating findings, I think, in this part of our work. I encourage you to look at our paper if you want to see more details, or to email me. I'm happy to discuss further, but I don't have too much time to go into this part today. But now we were able to use this atlas in order to look at the microglial response to immune activation. So here what we did is we injected poly-IC or saline, again at E12.5, and then looked now at microglia specifically, gene expression changes at E13.5 and E15.5. And broadly, what we saw is an increase in pro-inflammatory signaling 24 hours after the immune stimulus. I think this is to be expected. And then at E15.5, we saw a response that largely recapitulated what we had seen in other cell types, actually, which is a reduced translation and proteome synthesis and decrease in catabolism. And we ended up seeing similar responses across different microglial substates. And then we carried this out to postnatal day 14, which I expected to see a normalization actually of the gene expression profiles over 2 weeks from the stimulus. But surprisingly, we saw that some genes remained dysregulated or had changes in expression out this far. And this included a gene we're very interested in exploring further, which is HES1. We also verified the reduced gene expression by RNA in situ. And in recent years, there's been a lot of interest showing that HES1 plays an important role in decreasing the inflammatory response in microglia, in particular in response to a variety of different anti-inflammatory agents. So we're very interested in exploring this further. So to summarize, we saw that microglia are necessary for the typical response of other embryonic cortical cells to maternal immune activation, and that immune activation in utero leads to sustained transcriptional changes in embryonic and postnatal microglia. Of course, there's many caveats. This raised kind of more questions than it answered for us. So there's a lot of next steps. Importantly, I think for any similar study, transcription does not equal translation. So next steps include looking at relevance for protein expression and activity. We're also very interested in diving deeper into microglial substate specific roles and trying to understand, overall, is this response helpful or harmful in the context of in utero inflammation? Should microglia be there or not, basically? And can we harness this response for therapeutic benefit in fetal inflammation or in other diseases? And with that, I want to thank everybody involved with this study, especially my postdoctoral mentor, Paula Arlada, and Nuria, the second author on this study, who did a lot of incredible work, everybody else in our group and our funding. I'm happy to take any questions.
Great, thank you very much, Bridget. A reminder for the audience to please type your questions into the Q&A box. There's a button at the bottom of your screen. In the meantime, I'll get going with a couple of questions. So my first question is you mimicked a sort of viral infection in this study. Do you expect to see similar responses for other types of immune response, like allergies, for example?
So allergies is an interesting question. And from my understanding, a lot of allergic responses are more local inflammation and don't raise, in most cases, a large systemic inflammatory response. So in that situation, I wouldn't necessarily expect to see the brain respond in the same way. But for other infections, for example, bacterial infections, there's another model, the LPS injection model, which mimics a gram-negative bacterial infection. I would expect to see probably even stronger and more robust responses. And actually, we chose the poly-IC model because we thought that it would lead to a lower level, more physiologic inflammation that was more comparable to what was seen in the human.
Okay, thank you. My second question is, forgive my ignorance about this, but at what stage in the differentiation process are microglia when you administer that insult? Are they sort of terminally differentiated? Are they still maturing? Or what kind of, and is that important, the stage at which the microglia are?
So we think it is important, and we still have a lot to do about microglial, trying to identify microglial substate-specific roles. So we did do microglial sequencing at that same time point, which was E12.5, and we noted that there are some microglia that seem to have a more mature or maturing profile, but there are also a subset of microglia there that have an expression profile more similar to border-associated macrophages that actually makes up quite a large population of the microglia at that time. And we think they're probably a transitioning state coming into the central nervous system. And then there's a set of microglia that are actively proliferating that then quiet down over the course of development, with the exception of kind of a proliferative burst around birth. And so, yes, we think that absolutely the microglial substate is really important, and ultimately that we need to do some basically genetic tracing experiments to try to understand how more about the lineage relationship over time.
Okay, thank you. And we have time for one last question. So Kevin's asked whether there's another gene you can target other than CSF1R that would knock down microglia but not um, have the off-target effects that you mentioned for measuring those responses in mice.
Yeah, so there have been a few different approaches that have been used to knock down microglia, and I'll say actually the most common other approach is not another gene, but is oral microglial inhibitors, and so that's actually what I would suggest using in trying to understand more long-term effects of knocking out microglia is an oral inhibitor which includes inhibitors to CSF1R, but other receptors as well that you can temporarily ablate microglia, but then reconstitute them more specifically, and the mice survive longer.